Okay, so we have three fantastic speakers this afternoon who are going to present a variety of compelling topics in neuroscience for us to talk about. And first up is Olaf Sporns, who is joining us by Zoom from Indiana University. Dr. Sporns is a distinguished professor in the Department of Psychology and Brain Sciences and is the Roger H. Schaffer Chair at Indiana University. He's done an incredible body of work with him between him and his lab group in the area of neural network connectivity and how this shapes brain dynamics and function. And he's looked at this in the context of many neurological diseases, different neurological states, and across the lifespan. So he'll talk to us today about some component of that work, focusing on patterns in highly resolved fMRI connectivity dynamics. So I will pass it over to Olaf.
Thank you very much for that introduction. And just before I start, a brief preamble. I have been a little bit under the weather over the weekend, so my voice is not at full strength yet. I'll try to keep it up and see how it goes. Secondly, it was great to see the talks this morning or earlier this afternoon for me and see some of the history. And just to reiterate what Randy just said, some of us go back a long time, and Randy and I met first 30 years ago. It was in '92 at a workshop, one of the first ones on computational modeling of neuroimaging data. And so 30 years. 15 minutes is a very tough format to fill. I'll focus on just one idea that we've been pursuing in the lab for the past couple of years. There are many other topics I could be talking about, communication dynamics, connectomics, what have you, but I've selected this one because I think it dovetails nicely with the topic of this workshop. So that was just a preamble. This does not count towards my 15 minutes. The clock starts now. I'm going to start sharing my screen. Right. Well, as many of the talks have alluded to this morning or earlier today, one of the key problems in our field is to bridge between different modes of connectivity, especially the more static, unchanging skeleton, sparse skeleton of the connectome, the structure connectivity, and then the unfolding, rich, variable, flexible dynamics that we observe, for example, during resting state in fMRI, as you see in this little movie. And the question is, how do we get from one end to the other? What is the link? What are the causal mechanistic links between these two domains of connectivity? And I'll speak to that a little bit today. We've heard a lot about the virtual brain and about neural mass models in the service of modeling functional connectivity on the basis of a connectivity structure plus biophysical equations on top. And we and others have done this. Victor, of course, pioneer in this area, Randy as well. And we have also done a few modeling approaches in our lab that are more analytic, less detailed in terms of biophysics, but more graph-based. The bottom line is we can do pretty well overall in capturing the overall covariance structure of functional connectivity. Not perfectly, of course. We can talk later about why we can never get to a perfect match. And we and others have pursued this line for a number of years. One of the popular ideas that we came to through modeling is that functional connectivity itself is dynamic. The networks, the functional networks that we can measure actually change over time. And a popular way of approaching this has been through windowing and looking at fluctuations of network states. And one of the ideas that early on popped up in my head was that functional networks fluctuate between more integrated and less integrated moments in time, moments of functional segregation, moments of functional integration. And that notion has kind of, has become in some ways a popular notion in the field, for which there's much empirical evidence coming from the work of Andrew Zaleski and Michael Breakspear, as well as the work of Max Schein, who have employed windowing. We've heard about this in earlier talks today, to capture small episodes of functional connectivity and then look at how these windows compared across time and what kind of structure you could find in them. We also looked at windowing in our own work over the past few years. And of course, Randy and Victor and others employing TVB to some extent have indeed made inroads there as well. I'll be honest with you, I never liked windowing all that much because the windows still are considerably big, a minute or two in length, and there's lots of free parameters you have to select before you can actually get started with the windowing approach. Not ideal in my opinion. So last summer, a couple of years ago, some of us had an idea which actually gets us out of this conundrum of doing windowing and allows us to access, I think, fMRI dynamics at a much finer time scale, down to the single TR, basically, single 1 second resolution. And it goes like this. Classic functional connectivity is often based on Pearson correlation of node time series, the bold activations that we measure with the fMRI system. And what we suggested was that we could unwrap the Pearson correlation into moment-by-moment fluctuations, so-called co-fluctuation or edge time series, which are really the product of the two bold time series on each edge. The key thing is that the mean of all points in time of those edge time series is exactly equal to the functional connectivity. So instead of considering one measurement at the end, which is functional connectivity, we unwrap it in time and consider the momentary contributions to functional connectivity at each TR. When you do that, you get rasters of edges over time that have this interesting sort of a striped pattern to them. There are moments in time when co-fluctuations are particularly high. We were concerned initially because these are usually or can be indicative of problems in the MRI acquisition, unwanted physiological signal or noise. And we've spent quite a bit of time trying to chase down the potential sources that could give rise to these moments of high co-fluctuation. You can measure them or you can track them using simple summary statistics such as the root mean square. We colloquially refer to these moments in time when co-fluctuations are particularly high as events, which turned out to be somewhat unfortunate, but look, we can discuss why that is. We find these bursty event type fluctuations in really all data sets we've looked at, and they're unrelated. I'll tell you right now, they're unrelated to head motion or cardiac respiratory cycles. We've been pretty fastidious about excluding these sources of variance. Because this is such a robust effect, we were interested in where it comes from. I'll show you a movie here just to drive home what this looks like. You just take a regular bold time series and you compute co-fluctuations on each TR. And I'll set the movie in motion. What you see in the upper left-hand corner, that's your classic functional connectivity. The point is that the mean of all the frames that you see displayed one by one in the middle, the mean of that is exactly equal to what is shown on the left. So the construct on the left is familiar to all of us. We've talked about it all day. The construct on the right is the moment-by-moment contribution that the bold time series make to that construct. As you can see down here, the edge time series display this stripiness and event-like fluctuations. So this is really fMRI dynamics at a very, very fine time scale of 1 second, or in this case, 720 millisecond time resolution. Let me see here. Yep. One thing that you observe, I should have pointed out in the movie, is that functional systems are not equally well expressed across time. So the functional architecture we're so familiar with, and let me go back for a second, visual, somato motor, attention systems, frontoparietal, default mode, and so forth, that we're so familiar with from functional activity analysis is not well expressed at any given moment in time, right? You could see that in the movie. In fact, different systems come into and go out of co-fluctuation amplitude very quickly. You can perform all sorts of clustering analyses, and you can come up with distributions of how often systems are expressed and what combinations they occur. It opens up a whole new way of analyzing functional connectivity. So we were interested in what does functional connectivity look like around the events? Does it differ from what we observe when there's not a high co-fluctuation state? In fact, it does. In fact, it's driving what we're seeing in global functional connectivity. Event frames are much, much closer and much, much more quickly approximate the classic resting state network architecture we're so familiar with when we aggregate them together. And there's a common signature here. It's actually very similar to what has become popular as being called the principal gradient. We can discuss this later. I'm not a great fan of gradients, to be honest with you. But there it is, with default mode and fundamental systems on one side and sensory systems on the other side, co-fluctuating against each other. One of the things that I wanted to mention is there was a paper that came out as a result of our first publications that was actually very helpful in understanding where these effects came from. The robustness of what we observed was kind of puzzling to us. And sometimes when you have such large effects, it's actually because there's an underlying mathematical regularity. It turns out that indeed you can decompose covariance matrices, we've done it ourselves in our own work, into, let's say, eigenvectors or to PCA. And you can certainly find components in functional activity covariance matrices that will drive that structure and have a particular structure to them, which resembles what we see during events. So this paper here was actually quite helpful. Points to remember is that many of the findings that we published in our papers really weren't widely appreciated in the fMRI functional activity literature, despite the fact that there was antecedent work. And the math that Novelli and Razi provide actually, I think, confirms them and gives them some mathematical foundation. Edge time series are, I think, a neat construct because it's the same data you use to compute classic FC. There's no additional parameters needed, no windowing, no statistical manipulations at all. It's sort of really nicely organic in that sense. And unlike these analytic decompositions that Novelli and Razi provided, edge dynamics do have temporal specificity. So fluctuations like events occur in time at specific moments. And this comes home in spades when we look at movie data. For instance, we have a couple of studies now in submission in which we look at movie data. And when you look across subjects watching the same movie, you find that these event-like fluctuations actually line up across subjects. So they appear to be triggered or stimulated, or perhaps related to cognitive processes. A couple of studies now that I want to explore in my idea that I want to pronounce here. They're kind of related, and both of them are work done by two of my graduate students, Maria Pope and Sarah Kapps. The first one is, can we actually build a generative model for event-like fluctuations? The answer is yes, and in fact, we can show that these fluctuations that we see in real data are traceable to modules, to structure connectivity clusters that are driving these events and are responsible for them in a sort of causal way. Secondly, can we use these fine-scale fMRI dynamics to enhance or increase signatures, and for example, of individual differences across individuals? And the answer is we can. So first, about the model. Fairly standard, I would say, Kuramoto implementation, Kuramoto oscillator with time delays. And then on top of that, a hemodynamic response function. We've seen these models illustrated before, especially in Victor's talk earlier on today. And our question was, when we simulate these types of models and perform standard analyses, will we find fluctuations that resemble the kinds of events that we see in actual empirical data? And the short answer is we do. An example is shown here on the right. This is a simulation. And I don't have time to go into details. We can talk about it in the discussion if you have questions about how we actually do this. It's a fairly robust modeling. It's a phenomenon that pops out of the model without doing a whole lot of parameter tuning, really. There's only one free parameter, really, which is the global coupling. We were interested in where these events come from, because in the model, this is the advantage of having a computational model, we can actually manipulate the model in certain ways. One way that we did that is by taking event timeframes, clustering them, and then looking how they aligned with structural connectivity patterns, and specifically with structural modules underneath. And as you can see in the bottom row of this diagram, they align quite closely with those structural clusters or modules that are ingrained in the connectome, if you wish, that we provided. If you do away with these clusters by rewiring and producing or forming a random network, you will also get rid of events. This was fairly good evidence that indeed event-like fluctuations are in fact dependent on structural connectivity, more precisely on synchronization events that occur in the structural connectivity matrix, in these clusters, in these modules. So we can conclude from the model that functional connectivity, at least in the model, has a causal mechanistic origin. So it's not just sampling, variability, as some have suggested, but there's an actual causal story to be told. Nodes synchronize with each other on the fast time scale of Koromoto phase oscillation. And then this translates into low frequency signals that can be picked up with fMRI and are in fact have a spatial topography to it. Secondly, fingerprinting is a phenomenon was introduced by Emily Finn some years ago, and refers to the fact that if you compare individuals across multiple sessions of imaging, especially in resting state, you can find sets of edges that are stable across multiple resting state sessions, essentially constituting a functional connectivity fingerprint that can identify these individuals. There are methods available for how to do this, which we have appropriated for our study. And it has been known for some time that these edges are localized to, for example, frontoparietal attention systems. So they're not everywhere. And there has been preliminary evidence from our own work, but also from the work of Joaquin Goni and Yuko Miko, that the fingerprinting itself is sort of temporarily localized to brief episodes during the run. We wanted to use our fine scale fMRI dynamics to see if we can localize fingerprints, not only in space in these particular systems, which we could, but also in time. The basic idea was we take our edge time series, which we can decompose into frame-wise contributions. We filter these time points, usually retaining only 10% of them. We construct a corresponding FC component and then score them across individuals for fingerprinting accuracy, using a bunch of different criteria for filtering and using a bunch of different measures for fingerprinting itself. We then also employ an optimization strategy to identify optimal filtering strategies that enhance the fingerprinting signature. We did this on 2 different data sets, multiple measures, et cetera, et cetera, et cetera. And I'll cut to the optimization over here. Performing optimal performance, selecting a filtering strategy that gives us the highest fingerprinting accuracy, we can do much better, much better than using all the data. All the data is illustrated here by a stippled line. 10% of the data can give us a much, much better fingerprinting signature. Importantly, we can do this on a subset of the subjects, and we can then use held out subjects to verify whether the strategy actually works, and subjects that the optimization has never seen before, and it does transfer very nicely. So we can use a filtering strategy in time, single TR resolution, to identify moments shown down here when fingerprinting is best or performs best across individuals. We are currently engaged in an effort to basically employ this strategy for brain behavior relationships and see if we can enhance brain behavior relationships in this manner as we could enhance fingerprinting. I am almost at the end. That's actually the end for me. That was the basic idea I wanted to get across. What we can do with fingerprinting and modeling of fine scale fMRI dynamics. And I'll open it up to questions.
Great, thanks for a great talk, Olaf. I will open up the floor for questions. And again, those of you on Zoom, feel free to raise your hand or just type your question in the chat box.
Hey, Olaf, thanks for the talk. Awesome to hear you speak on this because I've been reading the papers and haven't been doing much for myself. I'm curious to degree, because this is like a great simple approach to doing frame-wise, but of course, as a lot of functional connectivity does, it relies upon simultaneity of signal in the data. I know the bold signal is sluggish and all that, but to what degree do you think there's room for looking at phase-based measures, even with this edge time series, right? And whether the bold signal even has the potential for phase to be meaningful?
It's a good question. My gut reaction is absolutely. This was for us such a new idea a couple of years ago. We spent quite a bit of time just sort of running down the basics and making sure we were not kidding ourselves and doing controls. And all the studies took longer than we thought they would. So we haven't quite gotten to the point how to go beyond this construct. And I'm a little reluctant to do that, quite frankly, because I like the simplicity of it. The fact that we do nothing to the bold time series. You just think about you can do this analysis using the code we provided with our publications in 5 or 10 minutes. You know, if you have a resting state one, you can actually do this and verify whether this works in your data as well, without having to do any sort of fancy quote unquote, you know, post-processing or manipulation of what goes into the time series or the relationship among them. Another thing that we have not explored yet is to do things like time lagged, like cross correlation. So you can sort of approach directionality if you wish a little bit, and we have played with it, but haven't gotten very far. So that's another direction that is worth pursuing.
Cool. Thank you.
So focus.
No idea where the camera is. I assume this is where the camera is. Well, thank you very much for this talk. This is Kalila Christo speaking at UBC. I have a question about these bursts, the events that you presented. I'm curious if you've thought about doing something that we've tried to do something a little bit around, which is to probe people. If you can do this analysis in real time during the fMRI scanner, you can probe people in a way that we've done to ask them to report on their subjective states immediately prior to the events occurring. So question for you is, are they detectable in real time? And have you thought about doing that? What do you expect they might report?
I think what is difficult to do in real time is to do all the post-processing that goes into getting the regional bold time series. You know, that's obviously something that requires, you know, cleaning of the data and doing all the stuff that we do on fMRI data, and that's not easily done like in real time. In real time, in fact, it's not really possible to do that. I'm very intrigued by the movie data that we've looked at. I was only able to show you just one summary picture, and there's a couple of preprints in play now. You can find them on bioRxiv that are actually dealing with movie data and look at what is happening in the sensorium, in the visual image, in the person's state, perhaps when you know, or right before when a large co-fluctuation occurs, and that to me is the closest we can get to trying to understand what possibly might be going on if there is a cognitive or perceptual sort of, you know, function to these things. In the simulations, there isn't, but to our best knowledge, these models do not have cognition or perception in the same way that we do. It's all dynamically driven, but a critical factor there is that you need that modular architecture. You need to have that. You need to have those clusters of highly connected, structurally highly connected regions that really, you know, that are found in fairly ubiquitously, I would say, in human and other species. And without that, you cannot get this. So I think there's a nice link to actually a very classic line of literature that's been going back at least 20, 25 years of cluster synchronization in statistical physics. And we sort of plumbed that literature now and have dug into it a little bit. So in the model, the origin of these things, it really depends on the architecture of the underlying structural connectivity quite a bit. And in reality, as it were, we don't know yet whether these states have any kind of cognitive or perceptual connotation.
Okay, we'll go to Victor first, and then Susan on Zoom afterwards.
Thank you, Olaf. This is Victor speaking. Can you see us actually? Can you see us?
Not really. I mean, I see the room, but I can hear you.
I'm the white one on the side here. So, okay, doesn't matter. Thank you. With regard to the H-time series, so when they, you cited the paper in Abufo et al. We have looked into this where the time series were measured in the human in simultaneous EEG and fMRI. So we also had electrophysiological activity there. where we computed the edge time series for the functional connectivity of the EEG signal. And I'm very glad to see your results. Also there we find coinciding increases of edge time series propagating through the network in EEG and in fMRI. And that was then later confirmed by Christoph also in the mouse. So I fully concur that this is real, the phenomenon that we have here. What we have found in the modeling is that this corresponds to cascades. So cascades are bursts of avalanches that propagate across the entire network. So our language earlier, across the manifold. And this comes out in the model data very, very clearly. Now, what you have done is you used phase oscillators. Yeah. So we have used population oscillators. And do you have any feeling whether you observe in your modeling the same type of phenomena, that there are these bursts of avalanches that we call the cascades that are propagating through the network? So these are individual events, very short time lived. Do you see this in the phasings also? And what are you feeling? What is your feeling about the meaning of this?
To answer your second to last question, I believe so. We have looked at, as you know, we get the full phase time series, which are sampled at 0.1 millisecond resolution in our models. And so we can do a lot of analysis on that, and we have done that. And indeed, we find that preceding large amplitude fluctuations in the fMRI signal, synthetic fMRI signal, we do see cascades, as you might call them, of phase synchrony spreading, usually first within and then between modules. I mean, the modular architecture is kind of really important. Your work, which ran in parallel to ours, is in some ways much more sophisticated, I would say, in terms of the underlying neuronal origin. I mean, your model is more detailed and more based in biophysics and so forth, and you've applied it to the mouse as well. So for us, this was actually great to see. I was a little skittish initially when we saw these phenomena in the simulations, because you never know what's, it's a simulation. You could be looking at artifacts that have a completely different causal origin. So I also think these are real now. I want to underscore this because there has been, there's sort of this weirdness going on with fMRI, because in fMRI, everybody thinks it's stationary, and all we're measuring is sort of a sampling. The fluctuations we see are sampling artifacts or somehow sort of stochastic things that have nothing to do with what's going on in the brain. And yet we know that the brain is clearly nonlinear, highly dynamic in the sense that you illustrated this morning, Victor, and of the finite time scale, we see rich fluctuations, of different size and size and length and duration. And so the fMRI field is still kind of, we still get reviewers that basically question whether there's anything dynamic going on in the first place. And it's just keeps puzzling me, because it's been puzzling me for 15 years. This whole business of windowing took off in sort of 2012, 2013. I showed people in 5 years before that simulations where we had windowed, and it's actually in the paper 2007 that there's additional timescales that you can get for free when you do these high dimensional neural mass models. And people just shrugged it off and didn't believe it was real or had any implication. And then 5 years later, all of a sudden, there it was. It's kind of weird. Anyway, I've forgotten what you said. Did I answer your question, Victor? I'm sorry.
Yeah, the question is, can we assign? I don't have an answer for this. Yeah, can we assign some meaning to the fact that there are cascades propagating through the network? And is that telling us something about potential for information processing?
Okay, so
I don't know. I want to see what you think.
I'm actually all over the place on this. I said earlier that we call these events, and I mentioned that I'm sort of a little bit, in some ways, this was perhaps a misnomer. Because initially, 2 years ago, when we first saw this stuff, and I remember I went over to your place, Victor, and like my last trip outside of the country before COVID hit was over to you, and we talked about it in Marseille. And what catches your attention are those events, are those sharp short-lasting spikes in co-fluctuation amplitude. However, this should not deceive us to think that that is the only cognitively important part of the run. Okay, I actually have a feeling it isn't at all. So one of the ideas that I was playing with early on, and I still am kind of fond of, is that perhaps these are just system resets. Okay, basically the whole brain kind of, it's sort of like a one episode of one chunk of mental operation has ceased. A new one is about to begin. Let's refresh. Okay, let's hit the enter key and start with a blank screen. Maybe that's what it is. So these really large events perhaps are just reset signals. This is one possible interpretation. In the movie data, they do coincide, at least one type of these events does coincide with gaps between movie trailers. And that's very interesting because that is when you would imagine people reset, right? They've just watched one trailer and now there's a little blank screen and now there's a new trailer starting, and that's when people show large co-fluctuations. The other thing is in fingerprinting, for instance, if you want to enhance fingerprinting scores, if you want to get better reliability of identifying individuals across different runs, you can do so with 10%. You can do better with 10% of the data than you do with the full data. So you can denoise the data if you wish. And one thing that you tend to get rid of is events. So it's not the events that make you, and I'm waving my hands here a little bit, that make you know, in the fMRI world, fingerprinting as being what it is. But it is actually moments in time that fall between them. This is very clear coming from our data. We've done this over and over again, and does transfer to held out subjects. I have no reason to believe it's an artifact. So events may not be the whole story. They may not be all that interesting. Maybe they're just reset signals. The other thing, my final point, they come in all different shapes and sizes. I mean, I'm sure you've seen this in your data, Victor. They kind of form a, if you look at either the amplitude or the magnitude of them, they form a kind of heavy tail distribution. So there's actually different types of events, number one, but then also there's smaller ones, there's larger ones that are somewhat more rare. So it's really a distribution of events that occurs. And it is not just the very tall ones we should pay attention to.
Before we move on, I just have a quick follow-up question to that. You might have mentioned this in your talk, and I didn't wrap it with reset events or potential reset events that
I think our question was just asked, but I could not completely get it.
They might want to take off your mask when you speak in the mic.
You hear me better now?
Yes.
The large reset or potential reset events that you're talking about, how frequent are they? If you're just measuring in a normal state, and how long do they typically last for?
Yeah, good question. You saw some of the pictures I flashed earlier. You could take another look at them in the papers also. It's not that frequent. Roughly speaking, maybe once a minute in a resting state session. That would be sort of my ballpark number. So in a 10-15 minute time frame, about 10, 15, 20 of them, the really tall ones. It gets to be tricky because again, there's a heavy tail distribution, so there is a long slope where you find, of course, then low amplitude occurrences as well. And again, I have steered my my students and postdocs a little bit away from focusing on events, to be honest, and rather take the entire spectrum of network states that we get by virtue of this edge time series decomposition, and treat it as if every time step is created equal. Do not pay too much attention to the amplitude, but just if you're doing let's say brain behavior relationship, we've just started that. Bottom line is, and stay tuned, we haven't figured this out completely yet, but the initial results suggest that we can increase brain behavior relationships in certain ways if we filter the data and focus on particular moments in time. So brain behavior relationships are actually differentially expressed across time, just as fingerprinting is. So I've kind of taken a more agnostic approach with respect to events and try not to focus on them as much as we did in the beginning, because they were very conspicuous. And the first thing we noticed, and in fact, it gave us a great amount of worry because with fMRI, as you know, there are lots of things can go wrong, and some, and will go wrong. And however, they are very robust, and as Victor's work has shown, they find them in his work. And of course, Victor, you've done it in mouse, right? And I know I have anecdotally looked in other species as well. In macaque, I see them there. I know Rick Betzel has looked at zebrafish, and he finds them in zebrafish. So it's in calcium recordings. So it's actually not, look, that does not look like they are completely artifactual.
Great, thanks. I will ask Susan's talk on Zoom. She says, nice talk, Olaf. I was wondering if the cognitive fatigue that is often reported by brain injury survivors, particularly those individuals who have mild injuries with little radiographic damage, could be due to a locking in, so an inability to switch in and out, a loss of temporal dynamics. Yeah, that's the question.
It's a great question. I have not personally worked with brain injury patients in quite a long time now, but we have looked at dementia. We have looked at individuals who have been diagnosed with MCI or Alzheimer's disease, compared them to cognitively normal controls. And again, it's work in progress, and it's hard to say anything definitive at this point. The differences are relatively subtle. Unfortunately, I was hoping for some big, big changes in dynamics or network states or distributions of network states, but it does not shake out as easily as we had hoped. I don't know what to make of that. There's a lot that one can, we have heard about windowing a lot today. I'll be honest with you, I will be quite happy if I never have to do that again, because I was very uncomfortable selecting window sizes and spacings and overlaps and padding at the ends and things like that. And then in the end, you get a 1 minute state, which is not really the time scale you're really interested in. I am, for the moment at least, quite enthusiastic about this really simple unwrapping that we're doing here. And I feel like a lot of studies that have been performed with windowing, looking at functional continuity dynamics in the past, could perhaps profitably be revisited using this relatively simple, straightforward approach. It doesn't require windows and just looks at these network patterns, instantaneous network patterns at single DR resolution. So I'm hoping that people might be willing to re-examine even things that they've already published to see if there's more and more signal that perhaps has been overlooked because of the windowing, the blurring that it induces.
Questions in the room or on Zoom? Okay, I'll ask a question then about fingerprinting approach that you talked about, Olaf. So really cool that you can identify individual signatures. A couple of questions about that. I mean, how many data sets do you need from an individual to be able to distinguish what you think is an individual fingerprint? And are there, I see that you've studied many different neurological states, different disease states. Are there certain conditions, diseases, age-dependent factors, sex-dependent factors? Are there certain conditions where this fingerprinting identification of these individual differences is most robust?
Yeah. So just to be clear, fingerprinting has been around for about 7 or 8 years now. It's coming from the work of Emily Finn. It's been published in 2015. In fact, there's a few antecedents in literature even. You don't need much more than 2 sessions. We basically have 2 resting state sessions and a bunch of individuals, and you basically try to, based on the fingerprinting signature, to identify individuals in the second session based on the first. And again, this is well established by now. And in fact, the spatial distribution of the relevant edges and nodes has also been published by Emily in her early work. It's from the parietal and some attention systems, not everywhere. So that's all been established already, underscoring what our contribution was, that we can actually do better with 10% of the data. If we score how well we perform in our fingerprinting task, identifying individuals across sessions, we can improve our performance radically if we cleverly filter out particular signatures of networks that are expressed at particular times. And that's sort of our contribution to this, which suggests that the individuality that's enshrined in the fingerprint somewhere is expressed at different moments in time at different strengths. And that was partly appreciated already by earlier work by Enrico and Joaquin, for example, also our own work a few years ago, but using windowing or PCA decompositions, which have problems of their own, you don't get the temporal precision that we can get with the single TR resolution that we've brought to the table. So that to me is our contribution. Fingerprinting itself, well established, wasn't our idea. And in fact, people are more interested now in understanding how individual differences relate to behavior. Fingerprinting doesn't really speak to that, it turns out. And that's where we've invested the last few months in trying to identify brain behavior signatures using our technique. But the sort of the early breaking news is that it looks like we can actually improve on that front as well by cleverly filtering out certain time points and not others.
Okay, great, thank you. More questions?
Thank you very much. Sorry about my voice. It's been plaguing me for the past few days. At least I got through it.
Thanks, Olaf.
Thanks a lot.